Good morning. I am Nagham Tabaja, a second year doctoral student co-supervised by Dr. Ilhem Benthaleb from Montpellier University Exam Lab and Dr. Lami Chalak from Lebanese University. We are working on monumental olive trees of Lebanon, drought history, pollution traceability, and lessons for the future. We will go through the state of art, objectives, studied sites, material and methods, results, and the conclusion. Why olive trees? They are significant symbols among the oldest trees in the Mediterranean, and in Lebanon they survived at diverse altitudes but not yet studied. Why the Mediterranean basin? Because it's an area vulnerable to climate change and human pressure. Species persisted in it due to low amplitude of temperature change, and the Middle East and North Africa region are major contributors to global health and climate change emissions. Some studies were made on olive trees. If we assume that olive trees are one of the best indicators of climate change in the Mediterranean, we can use them to reconstruct their past climate and environmental conditions in order to predict climate and human changes. The supposed age of the olive trees, based on their huge diameter, is not a reliable scientific data because the tree size is largely influenced by soil fertility and environmental conditions. The scientific methods able to date the olive trees and to understand their resilience are still needed, since the changes in water balance affect wood anatomy, annual growth rate, and isotopic composition. The stable isotopes can serve as valuable tracers of how plants today and in the past have interacted with and responded to their abiotic and biotic environment. In addition to that, isotopes in tree rings can be used as potential good indicators providing historical climatic data and ecological information. While the changes in carbon isotopic ratio in trees is often used as an indicator of the water use efficiency, which explains photosynthesis. For the nitrogen isotope ratio, during tree development it is less understood. Some studies showed that the tree ring N15 changes with tree diameter, which is mainly caused by the increasing rooting depth or the change in the N15 of the nitrogen sources exploited by the tree. Although we have a low concentration of nitrogen present in tree rings, which make it difficult to analyze, but still M15 has been measured in tropical tree rings, providing retrospective analysis of forest nitrogen status. For the oxygen and hydrogen isotopes, they are considered good indicators of temperature, precipitation, and humidity. The tree ring O18 can provide information of how climate changed over time. To our knowledge, only one study of olive tree rings isotopes is available, which was done by Rossi, who successfully used tree ring oxygen and carbon stable isotopes in addition to dendrochronological data in modern olive trees from Italy, showing that cross dating and synchronization is possible. The study of the growth rings of trees not only allows, according to the density of the wood, tree ring width, and isotope analysis, to evaluate the response of trees in a changing environment, but it also generates a high resolution paleoclimatic data. Second, the dendrochronology using trees' growth lines to assess their ages is widely acknowledged for many species. In addition, its combination with tree ring based radiocarbon analysis helps give more precise results. Wiggle matching method based on the comparison between the C14 curve and important number of samples have been assessed and allow to give absolute data. Several authors have warned about cross-dating errors because other studies in the same sub-region of the Middle East Mediterranean using radiocarbon dating on olive tree cross-sections suggested age less than 200 years old. In Spain, Arnan was able to assess the ages of monumental olive trees by counting the number of annual growth rings, reducing 627 years old. On olive trees growing in the Santorini area, Cherubini reported that the determination of the number of annual rings is impossible, but this was later refuted that the more recent age of Santorini eruption, based on C14 dating and counting, range between 1627 and 600 years before Christ, while this same work was acknowledged and corroborated by the geochronology and climate communities. Hence, olive wood dendrochronology studies can be a subject of dispute between scientific communities. We also see that there are new methodologies. We have the X-ray densitometry, which allows the measurement of the wood density variation at intra-ring level, giving data like the thickness of the late wood and early wood. This proxy is time consuming, costly, and destructive, and it also limits the widespread use of density profiling for tree ring analysis, hampering other analysis like isotopic analysis. While X ray computed tomography allows obtaining 3D density volumes that can be converted to large data sets of density profiles with high resolution to perform tree ring analysis. These techniques is not destructive, does not need sample preparation, and is costless. What are our scientific challenges? First, to detect the age of olive trees with irregular tree rings and cavity. Second, how these trees have and will survive the different shocks. And three, how will they continue to persist among upcoming. The main objectives to be achieved in order to study the environment and climate of monumental olive trees in Lebanon is 1. To assess the age of the olive trees, and 2. Construct the isotopic variation. Lebanon is located in the Middle East, surrounded by Syria from the north and east, and Palestine from the south, and the sea from the west. It is 10,452 kilometers squared, and it has a Mediterranean climate characterized with 4 seasons. We selected 2 main sites: Pshale in the north, with an altitude of 1300, with annual precipitation of 17-19 mm and temperature ranging between 5 to 22 degrees Celsius, and Kaukaba in the south, with an altitude of 672 meters above sea level, an annual precipitation of 746 mm and temperature ranging between 8 to 25 degrees Celsius. These 2 sites were selected due to their different locations with different climatic conditions in order to represent 2 different areas. We implemented the meteo station and the rain gauge with the help of Larry in order to collect rainwater and the temperature and precipitation on daily basis. We did sample collection on monthly basis from 10 trees per site for leaves and stems, and on seasonal basis for soil and litter. We also collected a wood sample from a third site in Tripoli for trial of shearing detection. We collected more than 200 wood cores taken from the inside to the outside of the tree, considering that the inside of the tree represent the oldest age of our samples. A first result was obtained by the help of NRA during the month of May, representing the C13 to N15 ratio between Kaukaba and Pshale for the different organs. These data showed that the soil had the highest relation, and it showed the variation between the different sites. These data will also be calibrated and compared to the climatic data, which is the temperature and precipitation, to study the climate. Other time series will follow to represent the whole 2 years studied. Another work was done on the mercury concentration, where we had a seasonal comparison showing that the summer have the lowest mercury concentration and winter needs more data in order to confirm that it has the highest. As we can see here, we selected only 4 trees representing the full area. Monthly variation of mercury concentration was also done for the different organs, showing that leaves have the highest concentration among the stems and seeds, while the soil have the highest among all samples. We also analyzed mercury concentration on 9 tree rings, which confirmed that wood have low mercury concentration. But as per other studies done on pine, it shows that wood can represent a larger mercury pool than foliage and bark in spite of its low concentration along a time scale, showing that the wood have double the size of mercury concentration than that of foliar. Now we go to wood cores. We took olive tree cores and tried dendrochronology in comparison to cedar trees cores taken from Lebanon. We can see that unlike cedar trees that has regular tree rings and visually detected tree rings that are countable and clear, olive trees does not have the same traits, where we have irregular tree rings and can have a lot of false detection. That's why dendrochronology could not suit our study. From there, we decided to compare the three different methods used dendrochronology, densitometry, and tomography, realizing that tomography was the best option we have, where it gave the best view of tree rings and the better realization between the true and false rings, which would help us select samples for C14 and isotopes. So we sent 40 samples as a start for. We still have some work in progress, where we have isotopic analysis for rainwater samples waiting for their results, Turing analysis using Fiji in process, isotopic analysis for wood cores after finishing the Fiji step, isotopic analysis for the 2 years time series of the different organs are being prepared right now, and the 90 new C14 dating that will be sent next month. In conclusion, the impact on olive trees can be detected through isotopic analysis. The results of this study will help us anticipate the response of olive trees to current and future climatic changes in both Lebanon and the Mediterranean. The olive trees are good indicators of mercury concentration and not yet deeply studied in the eastern Mediterranean. The wood rings have great potential to study the mercury concentration of the past even with lower concentration than that over other plant material. Using X-ray tomography and C14 can help build a reliable chronology for olive tree wood rings and study the isotopic variation and the radiogenetic variation in a more precise time resolution. Thank you.